Thank you, Professor Klinderman, for sharing with us an overview of this Herculean effort. So I'll invite everyone back for a discussion. We're now going to have a 20-minute discussion. As a reminder to the participant, we invite you to ask your questions using Slido. You can vote for your questions. That will help us prioritize which ones. So the question that got the most thumbs up so far is for Dr. Blish. The question is as follows. They want to know if there are specific chemokines whose expression correlate with disease severity. So you showed us some interleukins and some cytokines that were not very well captured transcriptionally, but we didn't hear from chemokines, which obviously are key at homing some of these cells in the tissue.
Absolutely. So honestly, I think my data is not, I mean, I went quickly through that, I will admit, the best to answer that, but a number of other studies like the big Yale study, I think would be a better place to look at that, where they had more like 200 patients longitudinally. And I believe IP10 was one of the key chemokines that was related with disease outcome and prediction, and which caught my attention in particular because I observed the same thing in HIV infection from data I did like 15, 20 years ago. So I would refer folks to many other studies that have, because to do that sort of thing, you really need ideally 20 to 50 people per group. And we are still dealing with small numbers. We decided to go deep in a few rather than broad in a large number.
Yeah, so for those that are not well versed in chemokines, I believe it's 6 out of 10, right?
Yes.
And so perhaps also some time points could help in dynamically mapping some of these factors over time. The second question is for Mazanifa. Got 6 thumbs up. Did you observe any batch effects between the 2 different sites where you tried to integrate your data between the Oxford cohort and the Cambridge cohort?
I mean, that's a great question, and I wish I could answer that definitively right now, because we are in the process of doing it. We think it should be, I think it would be okay, given the experience that we've had in the past integrating data sets within the Human Cell Atlas Consortium. Many of the sort of like COVID-19 publications where we've integrated large data sets, and these were healthy reference. But by and large, we have the WHO criteria for the patients, and they all fall within a similar kind of grouping, criteria grouping. So I'm very confident that we can, but definitively once we've done it, I can let you know.
Paul, would you like to add some thoughts? You are also part of a large group that is trying to integrate a lot of data.
So at the moment, the stuff that we've got coming out that UMAP I showed is all from a single site done at a, there were still potential batch effects just because you can't do that number all at once. But we've tried to minimize that with some sort of careful, hashing of the cells so that we're not, every plate had a good representation. We have, I agree with Mars, I think we can approach it with a reasonable amount of confidence because we've done a bit of this before. And we did try, at least at the beginning, to harmonize the SOPs so that the technical elements will be minimized. There are clearly other bits to control for.
So are you doing, sorry, finish your thoughts.
Right. No, that's that's the hope, but I mean, we actually have to do the integration and find out.
Absolutely. A question for you, Paul, that was raised. Do you see or think that there are functional variants in the blood? What are the proposed mechanisms for virus moving to the brain, heart, or other tissues within the context of the immune cells?
Well, that's a good question. I mean, you do find virus. I mean, this is not something we're examining deeply currently, but we have not found any transcripts, and we have looked quite hard in the white blood cell fractions that we're sequencing. So I think that is probably not although anything could have happened in the first 24 hours or so, but we just haven't got any evidence for that. You do clearly find viral sequences in blood during acute disease, although in our study we only found that in the most severely affected patients. So I'm not sure I can really offer any insights into that other than it's if it does happen, it's not sort of happening in front of our eyes. I would say it might have happened at a sort of earlier time point.
Do you think it's a question? Oh, go ahead, Miles, please.
I was going to say that's the same with our data set as well. We don't actually see the viral transcripts within the peripheral blood leukocyte data sets. It might be just a time of capture. That's a possibility.
Do any of you think that it could be related to multiple organ failures when patients are very sick with cell death that could, results in the release of some of the viral signatures we're seeing in the blood?
Well, if I may, I mean, we do see viremia, free virus RNA, actually, we can't even say it's particles, in, again, as Paul said, about 30% of the most severe patients. But like you guys, I mean, we looked in very severe patients and we've looked very hard and there's not a single transcript in the blood cells. So, I mean, I tend to think of it as that some free virus gets released if the lung breakdown is severe enough, and that might be what's seeding the other organs, or it happened. So I think it probably happens either very early or very late, and it's probably not mediated by white blood cells, but by free virus accessing a tissue it normally wouldn't access because it's normally a local infection.
Any other thoughts on the topic before we move to the next question?
I'd only say that, I mean, obviously there was a discussion of this in the last session, but if you look in tissues, it's also, you can get, a range of different things, but there are patients where, again, we've looked hard and we don't find any real evidence of virus, but that there's plenty of inflammation. So it, in the multiple organ pathology, it may, there may be different reasons for it, but it doesn't necessarily mean that the virus is packing the tissues.
That's very reasonable. We'll move on to the next questions, which are for Catherine. They're all around neutrophils. People are very curious. So you use a platform that allowed capturing the neutrophils. So the first question, I'm going to read it. Since it's unlikely that neutrophils are developing from plasmablasts, what is your alternative explanation for your trajectory analysis results? Can you also expand on whether did you explore the concept of doublets, for example?
Yeah, so I recognize there's been some chatter about this, and I want to be clear that we didn't claim that we'd proven a trajectory based on RNA-seq data alone, because that would be ridiculous. And we didn't also make any conclusions based on the UMAP appearance, but based on the trajectory analysis, which is intended to do trajectories. But it's definitely not a proof either way, and we're working on it. By sequencing the VDJ out of these cells. But first we need to figure out how to isolate them. And we have proven that in fact they don't survive cryopreservation. So this means we have to find sick patients, get fresh blood, and sort the neutrophils, which for anyone who's worked with neutrophils, it's not a lot of fun. So ongoing. And please read the paper and what we actually said, not what has been said we said. So as we said in the paper, we said that it is a possible explanation, but other explanations, many studies have seen emergency granulopoiesis, and that could be either bone marrow derived or via transdifferentiation. We cannot distinguish those possibilities without looking. But it is also an assumption that this is impossible, but there is valid prior data that B cells can transdifferentiate into myeloid cells. It's been mostly with macrophages. It's largely under the control of either PU1 or CEBP transcription factors, and we did see sequential induction of the master regulators CEBP D and E, which do control neutrophil development along that inferred latent time trajectory. Now, of course, that is only as good as the inferred latent time trajectory, which is only a snapshot in time based on spliced and unspliced transcripts. So we are trying to experimentally validate this, and it's just going to take some time because unfortunately for my area, but unfortunately for our science, our rate of severe infections plummeted. So it's been challenging, since we only see these in the sickest patients, to get enough samples at a reasonable time of day for the quick processing.
And just to expand a little bit on the neutrophil question, you show some differences across patients. And so there's a question pertaining to the technical artifact of it. Are we certain that the gain in neutrophils are not resulting from a batch effect, such as bias during purification by different hospital sites?
So, I mean, the neutrophil to lymphocyte ratio is actually one of the earliest reported clinical correlates of bad outcome from the reports out of China, first of all. So in general, there's been a lot of neutrophil, higher the neutrophil count and the lower the lymphocyte count consistently at every center in every country has been predictive of poor outcome. In terms of our study, we, because again, we're working with neutrophils, we only enrolled from one hospital, and the people processing the blood were actually my grad students and postdocs who were picking up the blood within 4 hours of blood draw and processing it in my lab fresh on the fly in groups of 2 and 4 to get the single cell running. So I don't think it's a batch effect. We have now, again, as I alluded to, we're working on about another 35 patients across the spectrum of disease, and we're consistently seeing this, but we're still, culling through the data. I forgot to address the doublet issue, it is in our published paper. If it was doublets, you would expect to see an increased complexity in the reads, so a higher ratio of genes to UMIs. And we did not observe that. In fact, the developing neutrophils were a little lower. It's also an incredibly rare outcome for there to be whole cell phagocytosis. Neutrophils generally don't involve in that behavior, even in the setting of HLH. It's mostly macrophages, but yeah, so.
Thank you for the thorough response. Common questions for all speakers, and I'll start with Paul. It's written, all speakers have, the question is around data access. So can you all discuss how you will make your data accessible, but also the raw data, the bind files, and the clinical metadata accessible to the community, as to provide a powerful data source for computational biologists to explore. So we've heard of you all discussing how you were thinking about merging your data. Can you, discuss raw data access and feasibility of it?
Well, I think hopefully Martin will say the same thing. So the national UK KIC and the national HCA effort in the UK is set up to do this through the Embl-Sanger axis. Additionally, for Oxford, because we've got other data sets, we've created a warehouse and a data access committee. And nobody's obviously asked for it yet because we haven't released it, but the principle would be that it would be freely accessible through that data access committee. I mean, the details of the access I don't yet know, but the guys who are running the warehouse for the data are really expert in this, and when we set it up, we prioritize that. So hopefully it will work smoothly, and it will be soon, I hope. So that's the principle. That's
wonderful. Muz, you want to add to this?
No, I think this is where it's an example of where the HCA infrastructure and kind of know-how will really come. Will be incredibly helpful, actually, because we've already established the data coordination portal, and then the sort of like managed access where necessary, and also the open access, and we can leverage all of those platforms with these data sets, which I think will allow people to actually get the raw. And also, and another way of disseminating data, not just the data, i.e. through the web portals.
Absolutely, and I think Catherine also. Yes, I was going to say, Catherine, we know your study has been shared on multiple portals, so you're definitely leading by example. Do you want to expand on how your data is accessible to the community?
Sure. I mean, not too much to say. We've shared it through the HCA portal, as well as it's on Import and on several other portals. And we shared early and plan to do the same with our next data set as soon as we've made sure that we've done the quality control and everything. We also are building a bigger portal at Stanford that will have more detailed clinical, de-identified clinical information, as well as the other assays that are being done at the same time in terms of Luminex and things like that. So hopefully it will all be integrated even better soon. But as with everyone else, we're still sort of building that integrated portal and making sure that works well. But all the raw data is out, so, and will continue to be as we feel comfortable.
Thank you all for sharing your data. That is greatly appreciated. This is a question, I think, for Paul and Catherine, because I think both of you are using SQL and 10x. The community wants to know, does the SQL data compares to the 10x data if you put aside, of course, the granulocytes?
Catherine, you could start, I guess, as you've done a lot more SQL on this.
I mean, they're pretty similar. We find that we actually get a little bit deeper sometimes with SQL, but we honestly, the reason we went with SQL for this study was, in some ways it's easier to batch because we were getting samples as they came in, 1, 2, 3, 4, 5 at a time. And the nice thing with SQL is that you can freeze the slide and come back to it later and do them in batches of 8 or 10. Whereas with a 10x, you kind of want to load your whole chip. And we weren't sure we would always have 8 at a time, just, for cost savings. And also because we were getting ready, honestly, when COVID hit, we were getting ready to do a big flu study. And so we just poured 50 arrays and we just used them for COVID instead. Like, honestly, I think they're both great and they both have their advantages.
Paul, is there anything else you wanted to add?
No, I agree. We were in the same position where it was for an HIV study, and we used the SQL. But our experience has been very similar, and the guys who supported it have been fantastic, and the data merge is good. So I'm quite happy with both. We actually try to do them on separate samples, so we haven't got like a head-to-head comparison, but that will be done elsewhere.
Wonderful. There's a question for you. How will you use the LPS data as a control for the COVID samples?
Yeah, I mean, I think one of the problems that we've got with when analyzing COVID-19 blood samples is what is actually just, inflammation, what is actually specifically viral inflammation, and what is specifically SARS-CoV-2 induced inflammation or changes that we see. And the many more perturbations that one could actually compare with will give you that resolution. So this is where the kind of where you've got the severe patients, or as Paul said, the kind of sepsis patients that are not due to COVID-19. That's kind of like a good control. But on the other hand, you've also got the kind of asymptomatic individuals. And intravenous LPS, surprisingly, does not really cause much of a reaction to the individuals receiving it. So this is almost like an acute perturbation, like you do something to the an inflammatory trigger without inducing sepsis or making them ill, and you can see what the kind of acute changes are, and that would be a nice contrast to COVID-19 asymptomatic and maybe mild patients. Right. So they're just spectrums to kind of.
A fascinating control cohort. We'll complete, I think, with at least one last question to all speakers. Do any of the speakers have a sense of how the earlier use of steroids in severe patients are working? What cells and circulating factors are changing? And to that, if you want to comment on any evidence of specific complement activation. So use of steroids.
I don't know that we've looked.
Yeah. I mean, because the trial came out from our the recovery trial, we had an early look at this, but we didn't, because of when we did it, we didn't have enough patients in our study to really make any sense of it. And I mean, they are very heterogeneous, unfortunately. So I can't answer that question, but we would love to do that. I think it's a really important one, because actually only 30% of people get the benefit. And so these sub phenotypes are going to be really important if we can assess that.
And none of the cohort that we have, it was more before the recovery trial, and they didn't receive any treatment. Just they weren't given steroids.
Did anyone observe evidence of specific complement activation in the blood? I see no. Okay. Avi, there's a very popular question for you. I am not sure if you can quickly come online. Oh, yes. So is there a role for the Human Cell Atlas to organize a meta-analysis of single-cell data from all of these amazing related cohorts?
If Fabian is on, he might want to get online as well for this. I think absolutely. We were waiting for, all of these studies have to first actually be conducted, just the experimental push to get this done under pandemic conditions. And then to do the first set of passes to just get something were done at such warp speed. Now that these studies are getting to the point that they're converging and coalescing, this is not just a hypothetical plan. This is very much a plan, and I would say that this is a plan that we're doing not just as Human Cell Atlas, but also in collaboration with other initiatives. For example, the Lifetime Initiative. Fabian, I think it's called Decode, the specific initiative inside Germany, because of course everyone, as they should, have jumped into the deep end of this pool and did the best that they could. And we're going to do this both across these PBMC studies that are in the immune system, as well as other studies. There are studies in autopsies, in BALs, and so on. So yes, there's a lot of work to be done, and those interested should definitely reach out, and I'll hand over to Fabian as well.
Yeah, thanks, Aviv. I'm really excited about so many of these nice studies with always sort of a nice addition angle come in. For example, the side-seq one we just heard, or then sort of adding trajectories on top of things. I do love trajectories. So I think really being able to leverage the original idea of, what I think initially Sarah and Aviv, and now we all hopefully envision to bring together many of those data sets and then use them for, let's say, association studies or just adding variation. I think that would be very, very exciting. And I think doing such meta studies turned out to be a computational challenge that Aviv and myself, most importantly, say our postdocs can tell long stories about. And I think being able to do this on that scale, I mean, it's just amazing. I'm really looking forward to that. I think once things are on bioRxiv, that would be a good time to get this started, right, Aviv?
I muted myself. Absolutely. It's just absolutely. If you think of the meta-analysis we've done of the healthy body in search for the cells likely to be infected by COVID-19, many of these studies started years before the authors contributed data, and people contributed a lot of unpublished data, but it was data that was QC'd and harmonized and annotated. Here, everything happened in 3 months, and people yet are in a similar place, which is just mind-blowing. Also indicates how far the methodologies have come, that we could jump into a new thing like this and yet generate data. And I think we're now reaching the point in the next couple of months where the meta-analysis can really kick off. We've also learned a lot from the previous meta-analysis done for COVID. So we're not starting from exactly the same place. And we also know, and that's a great benefit, that we don't necessarily need to start with raw data to perform meta-analysis. And given that this is human data and sharing raw data adds a whole layer of complication that could delay timing, I'm very optimistic about that as well.
That's wonderful. So with that, we're going to conclude the first session. Please join me to thank all the speakers. Thank you for having shared insights to all of these amazing studies, and we look forward to hearing more detailed results in the months to come. We're now going to take a 15-minute break. Please join us again in 15 minutes as we resume the second part of the session. Thank you.